A review of FOXP2 mediates sex differences in ultrasonic vocalization by rat pups and directs order of maternal retrieval by J. Michael Bowers and Associates. Some species are sexually dimorphic in terms of communication. The most prominent example in neuroscience research is the vastly different male and female bird songs of some species. This and other examples like it, however, focus on sex-based differences in communication to attract a mate. This research strives to address potential sexual dimorphism of offspring distress calls to their parent. If such differences are found, the study also strives to determine the cause of such differences and whether the parent's response is dependent on such variances. FOXP2 is a transcription factor. Transcription factors initiate and promote the transcription of certain genes. FOXP2 specifically is a member of the forkhead family of transcription factors, promoting and repressing the expression of genes responsible for differentiation and cell growth, specifically dendritic branching and axonal morphology, among other cellular functions. FOXP2 can cause speech impairments in humans, including but not limited to stuttering and language difficulties associated with autism. This role, however, is not unique to humans, as mutations in birds have also proved detrimental to song development.
In the materials and methods, brain tissue samples were obtained from rat pups and female and male humans, and Western blot analysis was conducted. Vocal emissions from PN4 pups separated from their moms were recorded for 5 minutes, and the average frequency and amplitude was collected. ICV injections were performed on rat pups under cryoanesthesia. FOXP1 and RPLI3A specific primers were used during RNA preparation for the siRNA treatment. Two-factor ANOVA, t-test, U-test, and post-hoc testing were used to analyze ultrasonic vocalizations. Regression analysis was performed on the human tissue. As seen on this diagram, FOXP2 protein was detected from the cerebellum, amygdala, cortex, thalamus, hypothalamus, hippocampus, and preoptic area, or POA, with t-tests for comparing males and females. The results showed that males had a higher FOXP2 protein than females in cerebellum, amygdala, cortex, and thalamus, as seen in this figure. There was no difference in the hypothalamus, or POA, as a p-value was less than 0.05. These regions are associated with reproduction rather than vocal communication. No FOXP2 protein was detected in the hippocampus for either sex. Cell count analysis was also conducted to determine if sex differences was a result of variability in the number of neurons expressing FOXP2 protein, but no difference could be found. This indicated that sex difference could be attributed to a reduction in FOXP2 in females. Male pups were found to have a greater frequency of vocal communication than females, almost twice as much as confirmed by the ANOVA test. The duration of each of this call didn't vary from males and females, with an average time of 60 to 80 milliseconds. Control males had 603 total vocalizations, while control females only had 350 calls. This was reversed for the rat pups that had received the siRNA treatment, in which males had 466 vocalizations and females had 465 vocalizations. ICV injections had no impact on the vocal production in either males or females. siRNA treatment was found to reduce FOXP2 protein levels, but there was no impact on the FOXP1 protein levels. To further investigate this, primary cells were cultured from PNO rat pups, where FOXP2 protein levels were once again reduced on the stratum and cortex. In another follow-up experiment, while a difference existed between the sexes and the control, there was no difference in FOXP2 protein levels in the siRNA and scrambled control injected males. Once again, females had a reverse trend where FOXP2 protein levels were higher in the scrambled control injected females, supported by this figure. When there is no dam retrieval, rat pups suffered from hypothermia, starvation, dehydration, or predation. Rat pups have evolved characteristics to adapt and avoid the separation. One example is the increased vocalization calls from males that would increase their dam retrieval over females. By marking individual rat pups in a litter from a nest, it was revealed that males were retrieved first before females, as shown in the visual here. For maternal retrieval, siRNA females were retrieved from before control females, while control males were retrieved before their male counterparts in the siRNA group. This indicates that ultrasonic vocalizations have an impact on the maternal retrieval. The figure on the left reveals that females had higher FOXP2 protein levels than males, while there was no difference in expression levels for FOXP1. Based on a correlation regression analysis between protein levels and mRNA levels specific to FOXP2, a positive correlation could be identified between the two, its results shown on the graph on the right.
Sex differences are established in the perinatal period when the brain is still developing. Because there is a centrality of FOXP2 expression in many different species, they were interested in whether there were sex differences in FOXP2 protein levels in the brain. They discovered that they were higher in males than females. The expression of FOXP2 in certain regions of the brain of females and males, as it's seen in the results, led to the conclusion that this is one way that sex differences in communication are initiated and maintained in the developing brain. Ultrasonic vocalizations by rodents are innate. When the rodent is separated or distressed, they can be modified by calling in response to predator detection or pharmacological treatment with anxiolytics. This indicates that sex difference in rat pups are also innate. Genetic factors evolved alongside certain behaviors that could be seen in the high versus low rates of separation-induced USV by rat pups. They have not yet found if the sex difference is genetically driven or a consequence of differential hormonal exposure during the perinatal sensitive period. The basis of an opposite effect of FOXP2 siRNA in females is not yet concluded, but they have concluded that it may reflect a rebound and subsequent overshoot in FOXP2 expression because of inhibition starting from a lower baseline. This will allow them to reach a critical threshold for a reactive expression. In rats, each sex is biased to a sex-specific maternal care in the higher rate of anogenital grooming performed on male versus female. This is behavior that is driven by testosterone-dependent olfactory cues from the males. There are advantages for the dam to favor the retrieval of one sex because manipulation of sex ratio in response to environmental conditions is used commonly by R-selected species that produce lots of offspring rapidly. Because siRNA-treated females were retrieved over siRNA-treated males, this goes against an olfactory cue mediating the dam's behavior. The fact that the anesthetized pups don't call and as a result were not retrieved by the dam helped rule out olfaction as a variable and supported the proposed hypothesis. There are gender differences in how the speech is articulated, such as the frequency, pitch, and the sound of vocal production. Before children produce their first words, they start with gestures. It is observed that on average, girls produce their first pointing gestures earlier than boys. Girls show a consistent advantage in early language and development. They also speak words and sentences at a younger age than boys. They have a larger range of vocabulary and use a greater variety of sentence types than boys of the same age. By the time the children are 4 years old, they have gained their basic aptitudes that help them with language development over their lives. They have observed human brain tissues from 4-year-old boys and girls for basal levels of FOXP2 protein and found that boys have a lower level of FOXP2 protein than girls in the brain. This is the opposite of what they observe in the rodent, but why this is cannot be interpreted until there is better understanding of the role of FOXP2. But they infer that higher levels of FOXP2 in girls and higher levels of FOXP2 in male rats is an indication that FOXP2 protein levels are associated with a sex that is more communicative. They concluded that male rat pups might be more effective in communicating using USV, whereas in humans the communication advantage of girls is well known. These findings are the first report of sex or gender differences in FOXP2 expression in animal and humans. They concluded that there are higher levels of FOXP2 in the cerebrum of males. FOXP2 is associated with FOXP2 and their experiment did not detect a sex difference in the rodent model. However, in humans, they observed that there were a trend in an increase of FOXP1 in girls over boys. FOXP1 has been associated with language since individuals of variations in FOXP1 show language acquisition delays, with the onset of the first word being 3.5 years of age and having a vocabulary of under 100 words by 7 years of age. In an adult zebra finch, the males have a higher FOXP1 expression than females, and that proves that FOXP1, like FOXP2, may be associated with more communicative sex. Gender is defined by how we perceive male and female. Gender is the most phenotypic variable. Difference in sex can influence how language is perceived and processed and how speech is produced. Due to the unexpected contrasting levels of FOXP2 in rats by gender versus human by gender, their research failed to establish how FOXP2 levels as well as FOXP1 affect human language development. Interspecies variance in the case of communication development and needs make it difficult to apply an animal model to this area of research. Because current methods to measure FOXP2 levels in a human without harming them are not available, experimentation is not currently plausible. With technological advances, a case study could be performed in which a male and a female were individually observed from birth with monitoring of their language development as a function of their FOXP2 levels.